Yes, can we start recording? All right.
Oh. I am going to share my screen. No, okay, it came. So it will be with the last part of my presentation. As I told, it's not connected tissues. The last part will be on molecular evolution. And of course, this is a very, very long process, and there are many things we need to talk and discuss. I will give brief information. I just focus certain part. So it's sorry, I cannot give most of this molecular evolution part. And those are the books that I used during the preparation of this presentation, beside of the articles that I referred inside of the presentation, of course. So I would like to start with genetic variation. As you know, if there is no genetic variation, neither genetic drift nor natural selection will be able to change allele frequency, because there will be nothing to change. So it's first of all, there is the need for allelic genetic variation. This genetic variation can be allelic level or genotypic level. What is the main source for allelic variation? Mutations. Source of genetic variation, there are many sources for genetic variation. There are, of course, many different mutation types I'm going to show you. Source of genetic variation for prokaryote, horizontal gene transfer. It wasn't right, actually. I'm sorry, saying prokaryote not right, because nowadays we know the horizontal gene transfer happening between the different eukaryotes, from the eukaryote to prokaryote. Okay. But it first identified in prokaryotes, so because of that, I'm saying that one. Okay, what is the meaning of horizontal gene transfer? Most probably you know, but just remember that meaning. Normal gene transfer happening between the individual of same species. This horizontal gene transfer happening between different species individual, okay? But this is not the case also nowadays. So another source of genotypic variation, exon shuffling. Retrotransposons also can create genotypic variation. Motif multiplication can increase the motif number and can create a new gene. And one of the common mechanisms for genotypic variation is gene duplication. That is, I'm going to give example for this one actually. Many genes, new genes arise from this gene duplication process. Okay, recall mutations. As you know, there are many different types of mutation. I won't give information one by one. I am accepting, you know, those information, just the small information. Can be at the gene mutation, can be chromosomal mutation, can be genome mutation. Gene mutation can be at the point mutation or the frameshift mutation. There are many different classifications of these actually. So this is basic one. Chromosome mutation can be deletion, duplication, inversion, or translocation. Genome mutation can be the chromosome number can increase the one or can increase the as a group like the polyploids, the calling auto or allopolyploid. I will give information about that because one of the main source for plant species. Plant speciation actually process. So here is about mutation. I would like to give this information. What we know about actually mutation, most of the mutation in genome happening in human genome we have around 3.2 billion nucleotides in our genomes. Most of the part is non-coding, and most of the mutation have been in that part is neutral, means that do not affect any protein, do not change the protein production or phenotype. So it's we can say it's the most of them neutral. So it's the mutation can happen in the structural part of the genome or regulatory part of the genome. Okay. Here you can see simple eukaryote and complex eukaryote transcription regulation part, this regulatory part of the gene. It's the simple eukaryote, it's very simple. Complex mutazoan has very complex elements that are responsible from the gene regulation. I will give you information that one. So mutation can be, as I told, at the structural part, means that protein coding site. At the protein coding site will change, of course, amino acid composition. And when the change amino acid composition, most of the times can change the protein or enzyme, hormone, whatever is coding actually. This is a prokaryote, it's eukaryote. As you know, it's just the brief information. Prokaryote have unfragmented the open reading frame. In eukaryote, we have intron that interrupt the exon section. So during the transcription, after the transcription, actually, we have a maturation process, just the splicing to this intron region. Prokaryote doesn't have this process. And this splicing process sometimes happening, error happening, sometimes not all the introns splicing from this part, and so is the new mRNA can have the intron, so it has its kind of mutation also in sequence. So it's a when an mRNA after the production in prokaryote happening in cytoplasm, in eukaryote happening in the nucleus, when prokaryote produced the mRNA, that time when started produce mRNA actually, protein synthesis starts in that time. For eukaryotes need to mature mRNA. After maturation stage, mRNA has to migrate to the cytoplasm, and the cytoplasm attached to ribosome, and according to their codon, of course mRNA in codon, the primary primary structure of the protein shape in here actually. So what we know about mutation, I need to time also, I need to see because sometimes I cannot. What we know about the mutation, you know, so we have 3 nucleotides represent one code, codon or anticodon according to DNA, mRNA or tRNA. The mutation happened at the first and second position of the amino acid, and let's say this first position, second position, this is the third position. When the mutation happened in first and second position, it changed amino acid. But the mutation happened at the third position most of the time, not all of the time actually, do not affect amino acids. So this kind of mutation also can accept neutral mutation. And here's, as you can see, AAA or AAG. Let's say the mutation happened here, is the A turned to G, but still as the coding the same amino acids, nothing will change, just the brief information. Regulatory change. Regulatory change, this kind of mutation happen at the regulatory part of the gene. It means that it can be the nearby site, the promoter enhancer site, it's calling the cis regulatory part, or mutation could occur within the regulatory element like transcription factors that are encoded far away the gene that reading that moment actually. It's calling trans-regulatory section. Okay, so we will discuss in here which type of mutation predominantly contribute to adaptive evolution, structural or regulatory. Inside of the regulatory changes, which one, cis or transregulatory mutation, which one is most important? So here in the regulatory changes, protein itself do not change. It's stays same actually, but the expression level of the protein change, because the regulatory part of this gene change. It can increase the expression or decrease the expression, or change timing can change the expression. So it is the effect of this regulatory change can be like that. Can be effect at the transcription section. One mutation at the promoter or enhancer or transcription factors can happen. Is this affect this transcription during the RNA processing? This kind of mutation can happen during the translation, mutation in the ribosome regulatory region, like that, and also protein activity. You know, it's after the primary structure of the protein. Protein will gain the allosteric, will gain the conformational changes, will have a secondary, tertiary and quaternary conformation, and then we'll have the receptor section, and the mutation can happen in this process. Those are all regulatory changes. So it's a pattern of evolution. So it's which one is structural pattern of structural cis or transregulatory changes varies depending on level of divergence among taxa. Evolution in different kind of population and over different or evolutionary time scale may result in selection of different kinds of mutations. So those are information is important. We can say which mutation is important, more adaptive, not important, all of them important if it's not neutral. It depends on their, of course, position. According to book, actually, about the species level, cis regulatory mutation altering the morphology are more common than the protein coding changes. So it's the cis regulatory changes account for a greater proportion of the expression differences observed between rather than within species. So this kind of mutation, cis regulatory mutation, kind of accumulates over time. So it's kind of important, kind of have more effective in the above species level, like the genus, family, and the others. When we come to cis or transregular, which one is more adaptive? Which one is most important? Actually, we cannot say it's the most important, that it's not won't be right, actually. We can discuss here. The position of the gene, of course, is important in the gene regulatory network. Doesn't matter actually which kind of they are, cis or transregulatory. It's one of the most important things, the position of the gene in pleiotropy, means that affect how many phenotype and how many and also where in the gene regulatory network. And but there is one thing that is important: cis regulatory mutation happen close by the promoter enhancer region. That mutation affect that allele, only that allele. But transregulatory mutation, those are happening. Those are the mutation that affect that transcription after the processing, actually. So happening far away from that gene. So means that it can affect both alleles. So it's when you consider the effect, transregulatory mutation effect larger than the cis regulatory mutation. This is one point we need to keep in our mind. And okay, so at the end, actually, that one, I will come at the last part. Also, we will have a conclusion for these trans and cis regulatory and structural regulation part mutation. All of them important, but depends on where the gene located, and also timing is also important. So here in this part, in the second part, I'm going to focus on the origin of new genes. Where do new genes come from, actually? That I mentioned about most of the new genes coming from the gene duplication. Also, the other factor, there is a horizontal gene exon shuffling, retrotransposon, motif duplication, and the gene duplication. What is the meaning of gene duplication? There is an example. There is a gene. It's this color represent this color, blue color represent the full function. After a duplication, as you can see, duplicated, new gene can lose the function. It's calling the non-functionalization. Can gain new function. It's neo-functionalization. It's calling new function, or sub-functionalization happening, means that partition of function between the gene copies happening. And most of the, I'm going to give an example for this steroid hormone evolution for this point. According to Baker, he published 2001, what he recorded, I'm going to focus these two hormone receptor. Those are steroid hormone receptor, glucocorticoid hormone receptor and mineralocorticoid receptors, MR and GR. Steroid hormone, as you know, lipid soluble hormone, and they have receptor in the cell. Here we can trace in here the figure at this part. This is a steroid hormone, and after crossing the cell membrane in the cross the cytoplasm, attached to a receptor protein that's inside of the cytoplasm, receptor and hormone complex move to the nucleus. In the nucleus, attached to the DNA, and it's calling actually this is transcriptome. One of the transcriptome factor, actually. So it's the effect increase, stimulate mRNA production, and with this mRNA production, the protein, hormone, whatever it's actually was coding, production will happen. So here I'm going to give information about this mineralocorticoid hormone receptor evolution. What we know about this steroid hormone evolution, as you can see in here, this ER, estrogen receptors. One of the most ancestral hormone receptors is estrogen hormone receptor. And what we need to know about them, this estrogen receptor almost not specific. More ancestral hormone is less specific, means that can affect from many different ligand. It doesn't have to be ligand. Hormone actually can affect from many different sources. For example, estrogen receptor can affect around 200 different chemicals or the things. For example, bisphenol is one of the pollutant, actually. So is the DDT, can affect from many. Here, when we focus on this part, those are the represent glucocorticoid and mineralocorticoid hormone receptor evolution. As you can see, those are in the same, very close each other. And according to theory, the mineralocorticoid receptor evolved by glucocorticoid receptor duplication, gene duplication, how it's happened. Another study, okay, here's the brief information. What is this hormone and receptor working? Of course, you know the receptor is for hormone. If there is no receptor, hormone will not work. Mineralocorticoid hormone receptor is aldosterone. Glucocorticoid hormone receptor is cortisol. Cortisol affects many different tissue actually, increase blood sugar and the others, decrease the serotonin, so it can cause the depression actually. What we are focusing here is actually aldosterone. Aldosterone increase the ion uptake from the kidney, nephron. So also increase the water absorption from the kidney. So in this point, we can say we can make a conclusion, means that this hormone important for tetrapod, for animal that have they need to keep water inside. Okay, they have the problem about you know, the water. The animal have in the fresh water or doesn't have a water problem, don't need to have this kind of hormone. So here, another study about this hormone actually evolution, neurochordate first steroid receptor seen in here, and estrogen receptor seen around 50 million years ago. Here's glucocorticoid receptor, these mineralocorticoid and glucocorticoid receptors. As you can see, the ray fin fish, there is glucocorticoid and mineralocorticoid receptors. Here, this study is very nice study. I recommend you to read, actually, it's very nice. Evolution of hormone receptor, complexity and molecular exploitation. What they did, they set up different experiments, including both, you know, phylogenetic analysis and also resurrection of gene, actually, the first gene. With this study, they try to understand the gene duplication process of mineralocorticoid. What they ask in this question, actually, in this article, what was the main question? According to the studies, mineralocorticoid hormone receptor evolved before aldosterone and evolved before the tetrapod or teleost. So it means that when the mineralocorticoid hormone receptor evolved, there are no need to keep water inside the body. So they were asking how it's happened, how without hormone, how receptor evolve, and how without hormone, how yes, receptor evolve, we can say. Okay, they designed many different actual experiment at the end. What they concluded, there is a ancestral receptor, okay? It's basal receptor. This ancestral receptor, what I told before actually, more ancient, more less specific. This ancestral receptor wasn't specific to aldosterone. It was affected from the aldosterone, affected from the cortisol, also kind of primitive form of the hormones, calling the 12-deoxycorticosterone, DCO. And so this ancestral receptor was affecting from all of them. Okay, it wasn't specific for certain ligand. They made a nice conclusion, nice actually experiment, including the resurrection of the gene. And what they found, here I'm going to summarize, has long been considered the tetrapod species hormone because it evolved later, because the organism that inside water don't need this kind of hormone and receptor, of course. Aldosterone is absent from the plasma of lamprey. I'm going to show you what is a lamprey and hagfish. Ancestral receptor is activated by aldosterone, but aldosterone is absent in the blood of the basal fish. So what they found, the ancestral receptor was less specific after the gene duplication. Maybe in here's the, yes, this one is very nice figure actually, it summarize everything. This is the first receptor, not specific, effect from, as I told, aldosterone, cortisol, also these 11 DOC. Is the time period 470 million years ago. This jawless fish, it's called hagfish, lamprey, also in here. First, this receptor, there were this receptor affecting from the both DOC, the aldosterone and the cortisol hormone. But that period of time, there were no aldosterone hormone, of course. And after the gene duplication, one receptor with the changing to amino acid substitution, and these active sites of this receptor changed and became specific to cortisol hormone. And the other one didn't change and became the aldosterone hormone receptor. But as you can see, aldosterone hormone just here in vertebrate evolved after a very long time period. This is the very nice actually example for the hormone and receptor evolution. In here, what they concluded that, do I have time? Yes. What they concluded that first receptor evolved for aldosterone hormone, then hormone evolved. And according to their conclusion, before complex shape, actually aldosterone and receptor complex, there is no adaptive evolution for the receptor because there is no hormone for the receptor. So for adaptive evolution, they concluded it has to be complex. It has to be both of them, enzyme and receptor. Okay, are we clear? I hope. I am giving small, very short example from each part, okay? Here I'm going to give information about genome evolution, pattern of genome evolution, and evolution of genome complexity and interaction between natural selection and genetic drift. In genome evolution, first of all, there is many constraints on genome evolution. What is the constraint that which part of the genome affect from the which part of the constraint? Actually, I'm going to give brief information. First of all, non-coding part doesn't have many constraints on genome evolution because they do not, most of the mutation in this part is non-coding, not affecting their the adaptive evolution. So it's accepting the this constraint on the non-coding part is not effective. Here we need to first understand the genome compactness of the organism. There are two different types. It's very general actually. There are, okay. Two different types of the genome, large and compact. Compact genome is a genome of the small the viruses, bacteria, also archaea, and some unicellular eukaryotes. Most of the part of this kind of genome consisted from the protein coding, RNA coding sequences. There is, of course, there is a non-coding part, but not like the genome like the human or mammalian, okay, multicellular organism. You can ask this question, what about pangenome of bacteria? Yes, also, including that pangenome of bacteria. They don't have much, non-coding part. Expansive, the large genome we have, multicellular and some unicellular, eukaryotes also, of course, have this large genome. And majority of this gene in this organism is non-coding, containing introns and transposon. So here I gave information about genome compactness and constraint. As I told, the organism that have small genome size, they have almost have the coding genes. So there is very high constraint on that coding part, but non-coding part do not affect. There isn't much constraint for non-coding part of the genomes. Okay, there are two parts of genomes. Prokaryote and some unicellular eukaryote has small compact genomes. Most of the genes consist from the coding section. Eukaryotes, like us, multicellular organism, have very large genome, and most of the gene consisted from the non-coding part. So there is a nice study. It's published 2003, Lincoln and Connery. I'm sorry. They made a link between the effective population size, mutation, nucleotide mutation rate, and they worked on different species genomes from the prokaryote to vertebrates. What they concluded that actually, when the genome size increase, effective population size decrease, and of course, intron number and the transposon number, repetitive element in the DNA increase. And they came up with a conclusion. When we go up in trophic level, this is a trophic level, this is the producer. When you go up, it's the second and third the organism start. Consumers. So when you go up, the body size of the organism increase. Most of the time, not all of them actually. Genome complexity increase. There is, they had an argument about this. Yes, I'm repeating. When you go up in the trophic level, the body size increase, also genome size increase, but most of the genome consisted from non-coding elements, means that not working. So why is this happening actually? They had a conclusion about this from their this graphic actually. When you go up also in the trophic level, the effective population size decrease. Means that the organism that have reproduction potential in the population decrease. In this point, what we know about natural selection and adaptation actually, when the effective population size decrease, natural selection will be less efficient. Mostly we will see the genetic drift effect. When the natural selection less efficient, means that organism can accumulate this non-coding part in their genome, so their genome size increase. So it's this is argument. We have very large genome, and most of the part is non-coding, not working. It's not truly right, actually. As I told, some microsatellite related to the cancer, some intron also can join the mRNA processing stage. So it's not totally right, but its main conclusion, I can say, they have an argument, they have a conclusion actually. According to the conclusion, when you go up at the trophic level, effective population size decrease. When effective population size decrease, effect of the natural selection decrease, effect of the genetic increase. Because of that, we have a lot of non-coding part, useless junk DNA in our genome, what they said. But of course, some organisms not fit this one. For example, parasitic species has a small have a very small effective population size, but have also small genome. Some protists also, ciliates also have very large genome size, but just very high also effective population size. So it's not fit to every organism. But anyway, we can use this as a model, like the null model in the Tajimani or Hardy-Weinberg the principle. So this we can use as a null model. And this part, I hope I'm not confusing you. I'm going to mention about speciation. And the speciation part, first of all, we need to describe the species, okay? There are three different species concepts. One, biological species concept. According to this species concept, two individuals of the T individual have to mate. When they mate and give the viable offspring, they are same species. This is biological species concept structure. The phenetic or phylogenetic species or cladocistic species concept. According to this species concept, the last branch of the in the phylogenetic tree is accepting as a species. This is mostly using for prokaryotic organism because it's not easy to define the species border for prokaryotic organism. The last one, phenetic species concept. According to that, the same species individual has to, it's the same phenotype, but there are also, of course, as you know, the cryptic species. So it's not very, effective way to describe the species. This is very nice cartoon actually, define the evolution. So we are here, what are, okay, this is you are descendant from your parents descended from grandfather, great-grandparents actually coming from. This is our family tree, not this one, okay? It is not produced from our father or grandfather. Also, they describe the evolution like that. This is not evolution. In the evolution perspective, fish didn't turn to salamander, to cat, to us actually. There are common ancestor, and we have actually in here. So here, I'm going to summarize your speciation mechanism. Each organism have different speciation mechanism, or each, taxa, I can say. Animal, for animal, outbreeding, depression, breakdown of co-adapted gene complex. It's calling Dobrzyński-Müller incompatibilities. And result reproduction isolation, and this can create new species. In plant, polyploidization, one of the main mechanism for speciation. And the prokaryote, as I told, horizontal gene transfer, but horizontal gene transfer, what we know nowadays actually, not special for prokaryotic organism. And what is the model of genetics? Genetic tree, natural selection, mutation is the models of genetics. We can ask this question, is there any speciation gene? And what exactly is actually in species? I try to explain actually species, but for some organism, explain species not easy, especially for prokaryote like bacteria, because they are collecting different genes from their environment. So they have core and pangenome complex. So it's not easy to identify species for bacteria. So we have three terms. Speciation, the process by which population become different species. Second, microevolution, result from evolutionary mechanism, mutation, drift, selection, natural, the other, acting within the population. And when you combine all microevolutionary process combined, it's create microevolution. So it's happening above the species level, okay? So it's macroevolution refer the evolutionary change above the species level. Here, the one of the mechanism, the animal speciation, sorry, I didn't mention the third one, the first one. Three scientists independently came up this idea. Because of that, we are using three names: Bateson, Dobzhansky, and Miller incompatibility. According to this, when the species isolated from each other, each has different mutations. I don't have time. Okay, each have different mutation. Each can cross different mutation. Some mutation, will be, the fit for this population will increase the fitness of this population. The other mutation increase the other population fitness. But when you bring these two individuals from different population after a while, let's say 1,000 years later, it will be incompatibility. So it's will be a breakdown, a hybrid breakdown. So it's means that speciation happen. Of course, it has to be mutation, drift, or natural selection for a long time. And in the plant, there is also speciation gene. I'm going to just brief information. Actually, the mutation that happen in the sperm or the egg can affect the fertilization process. When the affect the reproduction, when mutation affect the reproduction process, of course, can decrease the gene flow between different populations. So this can increase the speciation. We can say that this kind of mutation increase the probability of the speciation. So this is what I mentioned about. For plant, most of the you can create new plant species with a single generation with this polyploidy event. Actually, it's very easy for plant. They tolerant to polyploidization. Polyploidization even happen in different taxa. Actually, it's vertebrate. We have also polyploidization process, but in plants, it's very common process. There are different kind of polyploidization. It can be allo or auto-polyploidization. And as I told, many crops that we are using polyploid, we did this polyploidization, but doesn't have to be actually as a, it's the stimulated, also can be in the natural way. There is another process, of course, after the polyploidization, there is a process, diploidization process, means that polyploid individual decrease the chromosome number because they will have a problem about homolog chromosome compatibility. So it's during the meiosis, they decrease their chromosome number, but doesn't have to decrease the gene numbers. That is important. I'm going to show you. This is process. Let's say there is a ancient genome, 2N chromosome with a duplication, auto-polyploidization process, number increased 4N, and with this diploidization for a long time after, is the chromosome number decreased 2N, but not like the previous one. Here, as you can see, this is one individual, the other individual after the polyploidization happened, because there is no meiosis in here, as you can see at the beginning, 4 chromosome, 2, 2. At the end, chromosome number decreased to 4, but kind of the combination of all the chromosomes. So here also show this diploidization process, fast and slow diploidization process, how happening. It's one of the tool, one of the mechanism actually in the evolutionary way. And the trade-off between polyploidy and diploidy, of course, you know, the polyploid species have many genes that, for example, one of the most important I can say, when one gene copy is not working, they have many different copy of that gene, so it is easy to you know, compensate that gene, the gene that's not working. So they are, if they are asexual reproduction, the fitness heterosis cross from generation to generation cross faster, and because of the many gene, they have a very high increased enzymatic activity. I can say the last part. Do I have? Yes, I guess in the 5 minutes I can finish. Evolution of development. In this part, the last part, I'm going to mention about evolution of development. So the first we can say, what is the evolution of development? How can it lead to evolution of radical changes in body plan? How can different types of development changes lead to different hierarchical evolutionary changes? Here, the tree, it's a nice tree actually from the SpongeBob families. The trees, you need to focus on this first tree tissue appearing here. For example, a segmental exoskeleton appearing here, solum appear in the Hox gene. Let's say it's the features, a different feature appear different time of different branch. Here, it is nice article. Most of the new body feature in the evolutionary, in the tree, okay, appear during the Cambrian explosion. Most of the feature, body cavity feature, the other features have this orange, okay? It was new contribution. First seen during the Cambrian explosion. So what happened during the Cambrian explosion? Which kind of gene affected during this explosion? Why that period of time? This is the conclusion. This is the figure of who was he and Dean. Hierarchical evolution of animal body plans. What they offer actually? They offer a hierarchy of the gene regulatory network and functions at different level of development of the body plan. They gave this figure for this one. They gave hierarchical modular structure. They said there are, we have developmental genes, we have gene regulatory networks. Mutation happen in different networks, gene regulatory networks affect different, can create different feature. Here they mentioned about there are kernels in gene regulatory networks. There are plugins, input-output devices. There are gene batteries. When a mutation happens in the kernels, these developmental genes, when mutation happens in these kernels, this mutation can create the phylum level differences at body plan. When the mutation happens in these plugs in or input-output devices like the Hox gene, Those can create the class, order, family, body plan changes. And the gene batteries can create a species level changes. So mutation happen which level and which part of the gene regulatory network in the developmental gene, it's important. And this is the last slide. Yeah, okay, it's great at the time. Evolutionary changes within the gene regulatory networks. Developmental biologists have hypotheses that most changes within the regulatory network will be cis-regulatory, okay, promoter enhancer at the gene. The reason is that cis-regulatory changes would only change the expression of one gene. On the other hand, trans-regulatory changes are often overly pleiotropic and then thus don't occur often, but when they occur, they have profound effect, so they can change the body plan. What happened during the Karahan explosion, actually, those trans-regulatory mutation at the center of the gene regulatory networks, it's calling the kernels, create different phyla during the Karahan explosion. The mutation happened, the genes battery affect just only the species level differences, but affect at the kernels, the kernels that trans-regulatory changes at the developmental genes can create new body plan and new phyla that happened during the Karahan explosion. What they concluded that it's not easy to happen a mutation, this trans-regulatory part, but when they happen, then it happened. It's not easy to make another. It's acquire a very long time because it's have a profound effect on the animal body plan. So it was my last presentation, last slide. I hope you are there. Are you?
Yes, Hocam, thank you for your informative presentation. We have two questions actually, if you can come to chat quickly.
Okay.
So one is from Melike Kazak. Can you see it?
I'm going to first, going to open the chat part.
Yes. From Melike Kazak.
From Melike Kazak. Where is it?
It's a bit up. She says there is, for example, there is two distinct populations.
Okay. One is more adaptive to certain condition. You have candidate corresponding gene. What should be used to test this gene? It is more like population genetics or ecogenomics. If more adaptive, means that ecogenomics. If depend of the feature actually is it's the adaptive evolution is ecogenomics. Okay, not population genomics, not history.
Okay, thank you. And Zelal Özgür Durmuş, is there any evolutionary event in the animal lineage where the genome duplicated as a whole, not gene by gene?
As a whole means that genome duplication, this polyploidies is also genome duplication, okay? That I mentioned about, we pass both animal and plant also pass many genome duplication. So it's a whole, we have whole duplication.
Okay. Thank you so much, Hocam. That's all from us.
Okay.
And we will continue with Manolis Ladikakis' Extending the Evolutionary Theory